Johnson at Oregon State. We're going to get going again with our talks. We're going to have 15-minute talks and then leave some time at the end for Q&A. That being said, we're going to get started with Casey Achala, who's presenting fungal microbiome associated with grapevine trunk diseases in Oregon vineyards.
All right. Everyone can hear me? Good. So yeah, good afternoon. Today I am presenting the final report of our project. We're looking at the fungal microbiome associated with grapevine trunk diseases in Oregon vineyards, and it was I did turn it on. Yes, I did. Okay, I think it's working now. Let me try. So if I were to change it, or here, no, there. Okay, all right. Sorry about that. So yeah, it was a 2 years project, and Monica Hernandez, she was a postdoctoral associate in my lab, and she now moved on to another position. She's a research scientist at one of the biotech company down in California. And Dr. Posey Busby, she's an associate professor at botany and plant pathology. Next slide, please. So for those of you for whom grapevine trunk disease is a new concept, it's a complex disease complex, meaning that there are several different fungal pathogens associated with the grapevine trunk disease, and it is known as the largest group of fungus infecting grapes. And at least 133 fungal species has been associated with grapevine trunk disease complexes. And probably these names are familiar for you. Eutypa dieback, Botryosphaera dieback, Esca, Blackfoot. Those are some of the complexes within the larger grapevine trunk disease complex. And even within the Eutypa dieback, there are several pathogens involved in causing disease. And the typical symptoms would look like the dieback of the cordons, shorter internodes, necrosis on the main trunk, and the stripes on the leaves, also called as tiger stripe, or necrosis on the crown region of the vine, or even the root rot. So those are mostly soil burn. Okay. All right, so yeah, since this is a disease complex, so many different fungal pathogens are involved in this. And we had another study that was completed a few years ago that was funded by Oregon Wine Board. We're looking at more traditional culture-based methods in understanding what are the major drivers of grapevine trunk disease in Oregon vineyards. And we following up with that, we felt like we did a culture-based method, but we might be missing some of the important drivers of grapevine trunk disease in Oregon vineyards. So instead of looking at the individual fungal pathogens, we thought of looking it as a more like a fungal community. So can we do a microbiome-based analysis of the grapevine trunk disease and look deeper into the how complex this disease is? And the other objective was to explore different pruning, wound, and soil treatments that would affect the fungal microbiome of the wound and soil samples. So we collected samples from two main growing regions in Oregon, Northern Oregon and Southern Oregon. And in Northern Oregon, we collected samples from 15 vineyard blocks. Southern Oregon, we collected samples from 16 vineyard blocks. And we used the drill bits to collect samples. We collected samples from top, middle, and bottom of the vines. We pulled them together, and then we did the DNA extraction from those samples. And like I said before, so after that, we looked at the different types of analysis. For the earlier part, we used the culture-based analysis. We plated the samples, and as you see in the figure there, we were finding so many different kind of fungal pathogens. And within that, Some were, like very fast growing fungus, some were very slow growing fungus. So when there is a fast growing fungus involved, the slowly growing fungus might have missed. So that's why it prompted us to do the microbiome based analysis. We have already published this result. If you are interested in learning more, you can scan this barcode here. So next, we were doing the microbiome analysis, and it was funded by the Northwest Center for Small Fruit Research. So Jesse Zimmerman, he was a master's student. It was his master's thesis project. And after we extracted DNA, we did the PCR amplification of ITS, and then we used the Illumina MiSeq for sequencing of the ITS. And we had around 350 samples to be sequenced for this study. So this is the quick outlook of how the results look like. I know this is a very colorful, and yeah, we needed to fit in so many different taxa in this figure, and I think Jesse had to be really creative on putting all these genera together, and I think he did a pretty good job here. So these are the vineyards that we sampled, and that's the percent relative abundance of those genera that were detected from the microbiome analysis. So let's say for vineyard number 1, these are all the different genera that were present in that particular sample. So yeah, starting from the bottom there, the top one would be Alternaria, Aureobasidium, Botryosphaeria. So, like the name goes on. So yeah, again, we are looking at the fungal community, right? So we were detecting all kinds of fungal community in those samples. So teasing apart only the grapevine trunk disease pathogens from that data set, we were finding that the grapevine trunk disease causing pathogens were relatively low in abundance compared to non-GTD causing genera. So those are all the other fungal pathogens that were present in the grape microbiome, but along with them were the grapevine trunk disease causing genera as well. So those are all the list of the pathogens that can cause grapevine trunk disease. And it was reported, one or the other study from all the grapevine growing regions where the grapevine trunk disease is a major problem. We also looked at how these grapevine trunk disease were different in Rogue Valley and the Willamette Valley situation. So this pathogen here, Truncotella and Cymatosporium, we were very surprised to see that these were the most common ones, and pretty much in all the strata that we were collecting samples from, it was present in almost 100% of the samples in both Rogue Valley and Willamette Valley. So pretty much it was present throughout the vineyard. And Phaeochromonium, Phaemonella, and Inonotus, that can cause ESCA disease. Again, they were we found more on the Willamette Valley. Botryosphaeria and Diporthi that caused Botryosphaeria dieback, again, was found more in the Willamette Valley compared to Rogue Valley. And Neonectria that causing the Blackfoot disease was also more in the Willamette Valley. So when we're looking at the two different regions, pretty much Willamette Valley vineyards had higher percentage of all these pathogens compared to Rogue Valley vineyards. And all the other pathogens that are listed on the left side, they were present there. We were able to detect, but not to, not a lot of percentage of those pathogens, but they were still there. So comparing to the microbiome-based analysis and our culture-based analysis that we had done earlier, so we had a very good resolution of how the grapevine trunk disease pathogens were present. So it was one of our objectives to see if we can find other pathogens that could not be detected with the culture-based analysis. So even in the Botryosphaeria dieback, we were able to detect 4 different genera. ESCA, we were able to detect 6 different genera, and Cymatosporium and Truncotella from the microbiome-based analysis we were able to detect in almost 100% of the vineyards. But in culture-based analysis, we were able to detect, but it was less than 10% of the vineyards. So it was really helpful to do the microbiome analysis on those samples. So another interesting finding from this analysis was that, so now we're looking at the community, right? So we're not just looking at the fungal pathogens. So when we're looking at the community, how the other fungal microorganisms interact with the GDD causing pathogens. So these on the bottom are the top 10 most commonly present genera of the fungal species, and those are all the pathogenic genera that cause grapevine trunk disease. And all these boxes, the shades of blue are negative correlations, the shade of red are the positive correlations. And wherever you see the number, that means those correlations were significant. So one of the interesting thing to see, for example, OTU5, the Aureobasidium species, when that species was present, we were finding negative correlations with all the fungal pathogens, meaning that if a sample had Aureobasidium species, then we were detecting less of the percentage of the GDD causing pathogens. So, now it makes us think, can we use these organisms for our benefit? So there was something interesting. And also the other species such as Cladosporium, one of the species of Cladosporium, also seem to be negatively correlated with the GDD causing pathogens. So yeah, that was something interesting from this analysis. So summarizing, this subjective metabarcoding is an effective tool for examining grapevine trunk disease in the context of the microbiome. GDD-associated taxa are not the most abundant, but they are the integral fungal community members. Cymatosporium and Truncatella are the most common ones. But we do not know their role. This name may sound new to many of us who are aware of the grapevine trunk disease, but these genus have been reported to cause grapevine trunk disease in other parts of the wine growing regions. These were most commonly present, but what's their role in the grapevine trunk disease? Are those the individual drivers, or do they complement the pathogenic species? So we do not know, and it needs to be further studied. And from this objective, so like I said, Jesse did his thesis. We presented these results in 4th Microbiome Symposium in Ecuador this past summer, and also Plant Microbiome Symposium at Corvallis. And Jesse is preparing a manuscript, and we're hoping to submit that very soon. So moving on to the second objective, exploring how the different pruning, wound, and soil treatments affect the fungal microbiome on wound and soil samples. So it was started right after COVID restrictions were loosened, and in the first year when we were proposing to start, we could not because of the travel-related restrictions. In the second year, we found a commercial vineyard, and then we started this research, and then so we applied in a commercial vineyard the different practices that can be used for the pruning wound protection. We did the soil treatment, we did the pruning wound treatment, but unfortunately, because we could not do the artificial inoculation in a commercial vineyard, we could not isolate the GDD causing pathogens. So we did not really understand the effect of these pruning practices. However, based on the preliminary data, the thing that we wanted to understand is if we were using biologicals, they had to be established in the pruning wound, or even if we are applying in the soil, it need to be established in the soil. So we just looked at whether we're able to recover the trichoderma, that's the formulation of the Biotem. So based on that, we collected samples, we tested them every 2 weeks until 12 weeks. And from the pruning wound samples, it did not look very promising. So we were able to recover the trichoderma only from less than 20% of the samples for first 6 weeks after application. And from the broth samples, only one time after 4 weeks of application, we were able to recover trichoderma species from the samples. And when we looked at the weather data, so those arrows are the first one for the wind treatment, the second one for the soil treatment. And if we see the temperature there, it said the average temperature during our application period was almost about 40 degrees. And for the trichoderma to be established, it is recommended to be applied around the 50 degrees centigrade, so 50 degree Fahrenheit. So maybe, like under our condition, under Oregon conditions, the biologicals needs to be studied further. Yeah, there are several good products out in the market, but when we use biologicals, how do they adapt in the local environment? That's very important. And yeah, those 2 days when we were detecting, the temperature kind of went up, but we are not sure if that was the reason we were detecting, but yeah, still in low frequency. So what next? So taking together from all these studies and understanding the biologicals and everything, so we thought, we might need a region-specific biocontrol agents that's suitable to the PNW environment. So we proposed this for our upcoming cycle. So it is going to be another 3 years of project. Thank you for funding again this continuation. So we'll be looking at the native biocontrol agents for another 3 years. And with that, yes, yeah, I would like to thank you all, and also the funding agency, Pacific Crest and Results Partner. They were very instrumental in helping us with the sample collection, and sometimes even sending the samples to us, because with COVID we could not travel, and they were just sending the samples to us, and we're able to complete this research. And Oregon Wine Board and Rogue Valley Wine Grape Association for other aspects as well. With that, do I have a time for a question?
Yes, we do have a few minutes for some questions. Yeah.
Okay, good. Yes. Yes, so when we were collecting samples, we wanted to understand what's the, like general scenario of the GTD in our vineyard. So we collected samples from both symptomatic and asymptomatic vines. And yeah, because of time constraint, I could not have all the slides here. And we looked at, like different aged vineyards. We looked at how the symptoms was correlating with the GTDs. And the most concerning thing for me was, like even in the younger vines where we cannot see the symptoms, these GTD pathogens were present. So yeah, that's why I feel like, if we were to manage GTDs, we have to start early on. We cannot wait until we can see the symptoms on those vines. So.
Thank you, Achala. Excellent work. I missed in the previous part. Did you use sample in different AVAs in Oregon?
So we sampled in mostly northern Willamette and the southern Oregon. So yeah, mostly two AVAs.
And did you find any difference in the microbiome composition between the both regions?
Yes, we did. So most of the times, oh, hold on. Yeah, the pathogenic population, when we're looking at only the pathogenic population in the northern Willamette was higher compared to the Rogue Valley vineyards. So, but when we looked at the like entire microbiome, we did not find differences, because like then we are looking at all different kinds of fungal species as well. But yeah, just at the GTDs we're finding differences.
All right. Do we have any last questions? All righty. Well, thank you so much. Can we get a round of applause? Now we're going to have Alec Levin presenting, taking proof of concept to wide-scale field use, the OSU high-performance dendrometer.
Check. 15 minutes. All right. 30 seconds per slide. Thanks, everyone. This is a project that we just wrapped up. I am really just kind of the facilitator of this project. So I'll give you the disclaimer of anything that you really like about what I'm going to say today. It's not on me. Everything you don't like is my fault. I just had the research site and the treatments all laid out in the vineyard, and then I worked with this engineering group to build this little guy here, the dendrometer. Slide. So the group that really developed this instrument, the device, is a lab that we have on campus called the OPENS Lab, or the openly published environmental sensing lab. And just to read you their purpose here, the Open Sensing Lab is focused on developing environmental sensing projects and research. Our lab provides the tools necessary for students, professors, and researchers alike to develop tools used locally and worldwide by the ecological science and engineering community. And it's just a really rad little lab that we have in the college, mostly operated by undergraduate students building all these little devices and stuff. Irrigation scheduling. Oops, that's the slide. Sorry, I have notes here. Irrigation scheduling. I don't want to waste your time. It's very important for many, many reasons. Slide. There's a lot of different ways of doing it. You've also probably seen people like me show you slides like this before. You can measure the soil. You can measure something in the atmosphere. You can measure the plant. They all have various trade-offs and advantages, disadvantages, etc. Soil-based methods are pretty inexpensive. You can easily automate them, but you have really no idea what you're measuring outside of like a softball size volume of soil, and there may be roots there may be not. ET-based methods are pretty easy to use, but have a lot of error associated with them because they're weather-based and they're not really measuring the actual plant or the soil. Plant-based methods are sort of like the gold standard, but they're very labor-intensive. And so you got to send a human being out into the vineyard to like measure something. So that's kind of the thing that we would like to automate is plant-based sensors. Slide, please. Dendrometry is not new. Dendrometry has been around for a long time. This is something I pulled from Wikipedia. It's a dendrometer from like the 1800s. It's in a museum in France. You can use these things you can use dendrometry to measure all sorts of different aspects of dimensions of trees. And one of the things that we're particularly interested in is this seasonal growth and the stem diameter variations, or the SDV. Slide. And so the principle is basically, let me try and walk everyone through this. The principle is basically this, is that we have this sort of broken down into 5 phases here from dawn to dusk. Change in stem radius on the top left, and then sap flow through the stem and on the right top axis there. The dark black line is the stem radius. And so basically what happens in phase 1, that's kind of the nocturnal period, you get a recharge of the storage water in the plant tissues as the plant rehydrates overnight. Phase 2 is like a lag phase in between the rise in sap flow soon after sunrise, and that's when you reach the maximum stem diameter, and that's where you can see that peak of the solid black line. Phase 3 is the period of fast shrinkage of the stem as it's losing water and transpiration is really ramping up, and you can see that sharp steep slope down as the radius or the diameter of the stem is decreasing. Phase 4 is another sort of lag or changeover between the max sap flow and the minimum stem diameter. And then stage 5 here is when the stem swells up and sap flow becomes negligible. So you get this diurnal oscillation of the stem diameter, and that's what the dendrometer is measuring. Slide, please. And so you can see this here. Here's the variation, the stem diameter, and we can compute all these different numbers from this. So we have this diurnal oscillation. MXSD is the maximum stem diameter for the day. MNSD is the minimum stem diameter for the day. You can calculate a daily growth, DG, etc. MDS is the maximum daily shrinkage, and that's kind of the one parameter that we're going to be hopefully looking at more closely here in this work. Slide, please. And so here is a paper that was published, I don't know, 7 years ago on table grapes, which is showing a pretty nice correlation between the maximum daily shrinkage on the y-axis and the average stem water potential on the x-axis. And so the idea here is that the stem water potential, which we use a pressure bomb to measure, very labor intensive, but it's kind of that gold standard measurement, as I mentioned. We're relating that to this this sort of the amplitude of that oscillation. And if we can measure that amplitude of the oscillation in an automated way, then we don't have to send human beings out with a pressure bomb, and then we've solved the world's problems. Slide, please. The dendrometer that was developed by the Opens Lab sort of has a number of different innovations. Dendrometry, as I said, is not new. It's made of carbon fiber, which has almost a near zero coefficient of thermal expansion. And we have some data to show you that compared to stainless steel or other materials. Sometimes the thermal expansion actually can outweigh the oscillations or the MDS that I was talking about. And so we got rid of that. The magnetic sensor that it uses, which is this little green bar here, they don't actually touch the sensor. They don't touch each other. It's a magnetic sensor. It's 0.5 micron resolution, and it's got it's a frictionless mechanical system as well. The sensor, the stem moves like this, and it pulls on this spring, and so there's almost no hysteresis when it comes back and forth. I mentioned the magnetic sensor. It's got an LED indication system, which we really liked when we were in the field, because when my technicians would go out to do the regular pressure bomb measurements, they would walk by all the little boxes with the electronics, and they'd tap a button, and the light would light up like red, yellow, or green to tell you that the sensor was in the right position. And we put a temperature relative humidity sensor on it as well. All right. Next slide, please. Here's another shot of the device on the stem, strapped to the stem there. There's the spring here, and then this sort of wishbone thing is anchored to the back of the stem, and as it expands and contracts, pulls on the spring, and it moves that magnetic sensor. Apologies about the small font here. You can see the displacement is shown on the left in blue, and it goes from -5 microns to 5 microns. And so the displacement here is only going between, I don't know, -3 to maybe 1 micron, so a 4 micron range. And the temperature there is going from just over 20 C to almost 45 C. And so we're pretty much within the range here of when we would want to be measuring something in the field during the season, and we're really not seeing a whole lot of movement of this dendrometer. Slide, please. Here's a look at the box with the button and the little LED light. After a couple of years of doing this, we managed to have the data of like what it's one thing to go out there and press the light to see if you need to do anything when you're in the field, but sure would be nice if you could have it like automatically loaded up to the cloud or something. And we did that after the second year. Slide, please. And then this is just the little temperature and relative humidity sensor that hangs off the Pelican case. All right, project objectives. Next slide, please. We got 9 minutes. Build a bunch of them, put them out in the vineyard in a replicated irrigation trial that already exists. We're going to change the when we start watering. So this is an existing irrigation trial that I've got going on already. We just threw these sensors out already. We've got 5 treatments here where we're turning the water on at different times, just waiting longer and longer to turn the water on. And so what we ended up doing was we just put sensors on the early start, the T1s, and then the really late start, the T5s. Slide. And then the last two objectives, which we sort of almost got to, was to make a bunch of measurements, monitor the vine water status, and then correlate the dendrometer signal with the pressure bomb. Slide. Vineyard location is down here in Southern Oregon in the Rogue Valley, where the experiment station is. It was at a grower site, but very close by, just to give you an idea of how far away it is from here. Vineyard is planted in 2014, a bunch of other information there. One of the unfortunate parts about this site was that it was hard to get the vines dry enough. Slide. This is just when we decided to start the irrigation. So the T1 vines, the early start, we started them when the stem water potential was -0.7 megapascal, and the late start vines started at -1.5, so super unhappy. And then once we started irrigating, we irrigated everything at 70% of ETC. Slide. We had 8 reps of these 5 treatments, and again, we only put the dendrometers in T1 and T5. So this is just like an idea of what one of the vineyard sites look like with the treatments. Slide. So here we're showing the green is the late start T5s, and the whites are the early start T1s. And the numbers in those colored boxes are just the device ID numbers. And even though they're shown in two different cells, we actually put two devices on one vine. We wanted to have two devices on one vine so they could kind of check each other. Slide. And so you can see them here on the far right. You can see the two devices. You could see these little like umbrella hat things that they built for him, which was after doing it for a couple of years. And we're like, we need to sort of protect, you know, you work for engineers that like sit in the lab all day and it's like, man, these things are getting rained on and like sprayers coming through. So they built these little like hats for him that did their best to protect them. This is definitely not like production ready. But we hung the Pelican cases and the associated electronics just from the fruiting wire. You can see there, and there's a couple others in the back. Slide. Just an idea of, you know, how the vines look. The T1s look generally pretty good. The T5s look pretty bad. Slide. Stem water potential numbers. We did get the vines pretty dry, but we didn't get a whole lot of separation. But we did, I'll show you some production data that, you know, the treatments did sort of the thing that we wanted them to do. This is just showing stem water potential over time. T1s were started on July 13th. This is in 2013. And the T5s were started over a month later. And so the vines all got pretty dry, and we managed to somewhat get some separation in between the water status of these particular plants. Slide. Just some production data, or when we started, initiation date. You can see 3 years of data, and we got pretty much a similar response in the vineyard every single year. The T1s are always in July, although 22 is a very late year. The vines didn't get dry until, our control vines didn't get dry enough until the end of July, and then the T5s not until the end of August. Total amounts of water applied in millimeters here, and the yields in kilograms per vine. So we did indeed apply different amounts of water, and we did get different yields. The yields were a little bit lower in the T5s, and a big reason why we didn't see a huge difference in the yields was mainly because the water, even for the control vines, we didn't start irrigating until quite late. So most of the berry size had been reached. Slide. All right, so the data from the devices looked okay. This is one of them. And I pointed out this is from a T1 plot. And so one of the things that, if you'll notice, this is going from May 22nd until through the middle of October this year. Right in the beginning, there's kind of this weird like settling in period where right when it starts recording, the number goes way down and then it starts to go up. You can see the growth of the stem from the very far left of the figure up to the sort of apex. That's the stem actually growing in size. And you can see the arrows that I pointed out, those are rain events or when we started the water, and you can see kind of a response there. One of the other things that we ran into is we had and this is kind of a pretty clean one. We ran into this huge, like you see the huge like dip and then a huge spike that goes off range. And so we had quite a bit of this going on that the folks in the Opens lab were sort of have to deal with. We have to figure out what was going on. They developed an algorithm to clean this up. I'll show in just a hot second. And the other weird thing that and this is showing displacement, by the way. So it's all in microns. And you can see, okay, you know, when it rains, it spikes a little bit. And when we irrigate, it spikes a little bit. And then the fact that it's all green in the background is part of that LED function, which is normally we would see the light in the vineyard. But once we started recording it, we could get that data downloaded with the sensor position already like built into the readout. Slide. Here's the same data, but with the cleaning algorithm applied. And so the blue is the original data, and the dashed red line is the cleaned up data. And so they managed to figure a way to, you know, we don't want to mess with the data too much, but we want to acknowledge the fact that like that's not real, like when it shoots down and then shoots up. So that's what that looks like. And then, of course, I had this question, slide please, of, you know, are these data any good at all? And I was looking back at the one paper that I showed where it was that MDS is a function of stem water potential. And I'll get there in a second. So I just wanted to show these next to each other to see, like, normally what I would expect is that it would grow to some apex and then it would not change after that the diameter would stay the same and it might oscillate. So I found it strange that it came back down again. So I need to figure out what's going there. I mean, maybe the stem is shrinking. That's not impossible. I highlighted, I highlighted from the published work, the maximum daily trunk diameter And then the line, there's two lines there. One is the berry diameter overlaid with the maximum daily trunk diameter. And pay no attention to the other stuff in the background, but I will ask you this. Do you notice anything weird about the scale of the figure? You have a 3,000 millimeter trunk. So I don't know what's going on there, but the pattern is the same. So maybe, okay, it's a typo, but this was, I was looking for sort of a growth and then a saturation point, and we got kind of this shrinkage there. Slide, please. The good news is that, you know, at least on a closer inspection, the oscillations in blue of the cleaned up data really correlate nicely with VPD. So that's good. So, you know, plants are doing the thing that we expect them to do, and the device is picking that up. So that's a good news. Next. But that's about as far as we got. So we're cleaning up all this data right now. So as far as our project objectives are concerned, I would say we got through 3 of them. And then the last part is going to be a lot of work with the data, because we just pulled these out of the vineyard like a month ago. And we have 2 years of data to get through. So we didn't get all the way through. Next slide, please. So that's the next steps, is to continue cleaning up the data and analyzing it together, correlating the MDS with the SWP, always refinement of the device components. I want to redeploy these things in some new plots where we can get a wider range of water status and not as many. And then I think really, ultimately, and I hate to say this, I don't know if grapevine is a good species to do this in, because I've been looking a lot more at the literature, back again at the literature, and grapevine may not be the best species, but we have some potentials to do this like in blueberry and stuff, and it could be a really good further investigation there. So next slide, which is the last one. Everyone at the Opens Lab put in a ton of work on this. Like I said, I'm just the messenger. People in my lab and our industry partners, this work was originally, you know, it was in another experiment that was funded by the Oregon Wine Board, and then the Oregon Department of Ag, and then Northwest Center funded the development of the device. So thank you very much, and I have questions maybe if we have time.
We do have time for a few quick questions. Yeah.
Walt.
One of the things to think about is with this device, you are applying some pressure. So you're going to have compression of the material that could cause of the plant tissue that could cause that decrease. And then later, as you get lignification and sloughing off of the outer part of the cane, you also will get shrinkage in that.
Yeah, that's a good point. And we try to, when we put it on, you know, early in the season, we try our best to scrape all the bark away and stuff, but you know, that's just the one season and it's perennial.
Right, but you've gotten woody tissue forming that's actually going to be shrinking, where you have the cambium layer that's going to be expanding.
Right.
Any other questions? All right. Well, thank you, Alec.
Thank you.
All right. So next up, we'll have Alex Wong, who's presenting Botrytis bunch rot, where, when, and what to use.
All right. Okay. Can everyone hear me? All right. All right. Thank you all for being here today. And I'm going to be talking about the Botrytis project that we've been doing through the Small Fruits Research Center. This project was also partially funded by the Oregon Wine Board, so thank you to them as well. So really quickly, I guess this needs to be closer. Just a quick intro on botrytis. It looks like we're missing part of my slide here, unfortunately. But bunch rot is typically caused by botrytis. I'm purposefully not putting in the species there because it can be caused by a disease complex, but is typically associated with botrytis cinerea, which you can see here on the slide right in the middle. And this is typically how we think about this disease, is actually a rot pathogen. If my slides had, I guess, transferred over correctly, there would be a picture here on the right of a like baby grapevine stem with a pretty sunken lesion of botrytis there. So it really is an opportunistic pathogen, and it would be eating any weakened, dead, dying plant tissue there. And so I'm also going to do a quick update or refresher on botrytis life cycle, because that's pretty important to how we manage this disease. So in the spring, conidia are produced, so the spores of the fungus, and they'll actually colonize the flower tissues. So if the flower tissues aren't dislodged from the cluster, they're going to be, they're going to die. And if it becomes colonized by botrytis, the botrytis will continue to eat this plant material. Later on in the season is when we're actually going to see symptom development, though. So in midsummer, these pieces of the flower of the inflorescence are still remaining inside the cluster. And once the grapes start to ripen and conditions become more favorable, that's when you're actually going to see pathogenic growth. So it's switched from growing as more of a saprophyte, eating dead plant material, to actually growing pathogenically. And then to restart the cycle, all of the dead plant material, botrytis will form things like sclerotia, and that's how it overwinter before the next spring, when it produces more conidia again. So the first question is of when is Botrytis inoculum present in the air? So historically, there's been a lot of spore trapping for powdery mildew in vineyards done by Revolution Crop Consultants, and have these spore traps, which you can see in the bottom right, which are spinning impaction air samplers. So conidia would in the air might stick to these rotating rods. And so this has been done for powdery mildew, as I said, but we wanted to go back because we held on to all of these DNA samples and reassess them for botrytis. And so we looked at 8 different sites over 6 years in the Willamette Valley and reran them for botrytis using a qPCR method. We also collected some of the more regional climate data, like precipitation and relative humidity, to see if there was a correlation between inoculum and weather there. And so what I'm going to show you is a kind of complicated graph, but I'll walk us through it. So each of these panels is a different year, and we've averaged the amount of botrytis from all of our sites in all of those years together. And so essentially what we have on the y-axis is your normalized amount of botrytis, so relative to that site within the season. So the higher it is, just the more botrytis there was in the air. And then on the x-axis is the Julian day, so what day of the year. And just as a reference, day 170 is June 19th, so pretty close to the summer solstice. Because phenology can vary pretty greatly, I just put approximate phenology up here in the different shades. So gray is just shoot growth, red is bloom, and orange is around fruit set. And so what's interesting to take away from this is that we do see these interesting surges in Botrytis inoculum around this time at bloom to fruit set. And so this is when the plant is also most susceptible, right? This is when there's going to be the flower debris from inflorescence that may remain lodged in the cluster. And that's where, Botrytis is seemingly most present. Now, this didn't happen every year. So in like 2019, in the top right, you can see that there was that spike a little bit later, but this was still before the cluster has closed off. So there's still this access of Botrytis inoculum to infect any dead inflorescence material that might have remained. So we also wanted to know, so just as a summary, Botrytis inoculum was pretty much always there. In 99.5% of our samples, we had a positive botrytis detection. And this was over 1,200 samples that we assessed. We saw these increases in airborne inoculum around when the plant would be most susceptible to colonization. And this did tend to associate with some of the cooler temperatures between around 55 to 70 degrees Fahrenheit. So in conjunction, we also did a sampling survey of botrytis. We sampled around 15 vineyards from both Oregon and Washington. Our sampling would start at around bloom and continue all the way through till just before harvest. And we would take these clusters and also any debris that we were finding on the vineyard floor, as well as any blackberries that might be adjacent to these vineyards, because they could be harboring botrytis, and we sampled those as well. We'd bring them back to the lab, incubate them, and then actually assess them for botrytis and collect any conidia for fungicide resistance testing. So this is sort of what we found throughout as a pattern throughout the season. So we did find that botrytis varied from site to site and year to year, likely to due to difference in disease pressure in the year and the microclimates of different sites. But we did see things like this. So on the left, you can see that right in that red circle, there's a bit of the inflorescence material that got stuck in this cluster, and it's sporulating with botrytis. So this would have become a botrytis infection later in the year around harvest. And so this image was taken in late July. On the right, you can see sort of the same thing. So this is in late July again, and we're already having rot in one of these clusters starting from inside the cluster. So this leads us to believe that, this was likely started way earlier, this colonization of the cluster, and would have been better managed earlier in the year. So we also looked at debris. So debris that we looked at was the rachis from the prior year. This image was taken in mid-June in 2021, and this is exactly how I found this piece of debris from that year. Is exactly how I picked it up off the ground, heavily sporulating with botrytis. Now, in general, botrytis on debris declined throughout the year, so it would be pretty high around bloom and then be much lower by the harvest, but we were seeing a lot of it. But what's interesting is this date. So we saw this around June 14th, so mid-June, and if I can bring your attention back to that chart that I showed earlier, in 2021, on June 14th, that red line in the middle shows about where this, when I collected this, and it's right during that spike and when we see airborne inoculum. Now, there's probably multiple sources of botrytis coming into the vineyard, but there does seem to be this correlation between the debris and when we're seeing airborne inoculum present. And then finally, really quickly on the sampling survey, we also looked at blackberries that may have been adjacent to these vineyards, and we sampled those as well. We found botrytis at varying levels throughout the season on both the flowers and the fruits. And so these could be a contributor of botrytis inoculum as well. And you can see on the right, there's a whole fence line along this vineyard that has wild Himalayan blackberries. Okay, and then the final part of this was looking at fungicide tolerance. So to date, we've sampled over 100 isolates, or phenotyped over 100 isolates to fungicide resistance, and we've tested about 7 or 8 fungicide groups. And our main findings is that we've found fungicide tolerance to all of the groups we've tested so far, and about 10% of our isolates have been found to be tolerant to 2 or more fungicide groups. And so that's a little bit concerning, and that we should continue to monitor for fungicide resistance. So just in conclusion, botrytis inoculum is pretty much always present, so it's something that we're always going to have to think about. And that if we're going to be managing for botrytis, it would likely benefit from managing at the most critical time point, which is going to be from about bloom to berry touch, when we're going to have these susceptible parts of the grape that could be colonized by inoculum that we know is going to be present. And so if we're going to spray fungicides to manage botrytis, we're going to have to also be thinking about what modes of action we're using. So if a mode of action is used, it should not be used for the rest of the year. But we should also be thinking about modes of action between years as well, because we're seeing a lot of this inoculum on debris, which was carried likely from the year prior. So fungicide tolerance could be moving from year to year as well. So a lot of care should be taken in selecting botryticide modes of action. So just in general, to wrap it up, botrytis management isn't going to be just, a single management strategy. It's always going to be a suite of things, making sure there's good airflow throughout the canopy and the crop, reducing sources of inoculum, monitoring the weather. If there's going to be a cooler, wet period during bloom, that's probably going to be a higher risk of infection and colonization of these clusters. And then finally, of course, using fungicide applications and using them when they're going to be most efficacious during that critical time period of bloom to berry touch. With that, we have published our results online. I'm sure you've all scanned enough QR codes today, but if you want to scan another one, we do have all of our results published online at this website. And we're going to continually update it as we continue to phenotype more and more botrytis isolates. And with that, I'd like to once again thank the Northwest Center for Small Fruits Research, Oregon Wine Board, all the grower cooperators who gave me access to their fields. It's greatly appreciated. Revolution Crop Consultants for sharing the DNA samples for us to process, and of course the foliar pathology lab at the USDA. And with that, I'd be happy to take any questions. Thank you. Yes, Inga.
Are there any fungicides that you would absolutely recommend to stay away from based on
So we tested a bunch of them. One of the interesting ones was Benamil, which obviously hasn't been used in grapes in a very long time. We did see a decrease in the amount of resistance, which is interesting, which might suggest that there has been a fitness cost, but I don't know if that would suggest that we should try to reintroduce Benamil back into grape, because likely it would just select for that population and it would go right back to being, ineffective for most of the population again. Yes, Chala. Are you talking about the inoculum, the debris? Yes. So as the year went on, it would become harder and harder for us to find this debris on the vineyard floor, likely because it's just getting broken down by mowing, or it's just decaying naturally over time, and that's why we were seeing this decrease in the amount of debris. Right, but the most key point is still that bloom to berry touch, and we were showing that we were finding quite a bit at that time point, and that's when it seems to be producing the most inoculum. So getting to it and managing it before we get to bloom would be the most effective way to manage that. Yes, Christian. So we've only looked at IDing botrytis down to species, and for some of our isolates, I believe we've only successfully or confidently ID'd 5 isolates so far, and one of them already seems to not be botrytis cinerea. It looks like it might be a botrytis that's found more commonly in floriculture. So that was an interesting finding. But we're still working on, doing more IDing of, the species of botrytis that we're finding out there.
With your 100 isolates that you screened, were they all from 1 year or were they from multiple years?
They're from all the years of our study.
Did you see any change in the resistance levels over the years? Or any shift?
Not off the top of my head, I don't think so.
Okay, thank you.
Any other questions?
Maybe.
Do growers try to control the botrytis on the vineyard floor with any sort of cultural practices?
I think the only thing would be like tilling to turn over the soil and try to bury it, but I don't think that that's something they intentionally do just for botrytis. So I would say no. It's just something that happens inadvertently when mowing or tilling.
All right. Well, with that, thank you very much, Alex.
Thank you.
And next up, we have Devin Ripner, who will be presenting the effect of soil parameters on plant parasitic nematodes of wine grapes in Washington and Oregon.
Hi, everyone. Thank you so much for the invitation to speak today. I'm subbing for a postdoc that was in my lab who did this research, a really awesome scientist named Catherine East. So I'm from the Horticultural Crops Production and Genetic Improvement Research Unit here in Corvallis, but I'm located at the Prosser worksite. And so let me dive into this. Let's see if I can. Okay, we'll just flip through it. I should have some jokes, right? Like that's the key here, is to be able to just like entertain people, because technology issues happen. We all know that. So yeah. So why could the A horizon of a soil and a B horizon not have any kids? Because they weren't parent material. That kills at soils conferences, but. So I'll just like talk through this a little bit since I'm here. So this is a beautiful vineyard in Eastern Washington. It's near Mattawa. And so you can really see kind of the soils that we're dealing with in Eastern Washington. I find this to be a great example, right? Very coarse textured soils. Here you see an irrigation line, right? And the soil has blown right up over it. No problem. And so these soils are very different than most folks are used to. Typically very high pH, right? 7 to 10. I've been in vineyards with pH 10 soils. For perspective, when I was in graduate school, if you had a pH 10 substance, that was hazardous waste. You had to bag it up and send it to environmental health and safety. And yet, we grow amazing grapes in these soils. I'm very grateful to Inga and her lab for all the hard work that they put into growing tomatoes for these experiments, for running these experiments. None of this would have been possible without her, right? And so I'm very grateful for the work of your lab, including Amy, who, is a real asset to the unit. And there's Catherine up there. We're at Lake Chelan. That was a fun place to go to. If you ever have a chance to check it out, it's definitely worth it. Yep, that should be it. Okay, let's see. Oh. There it goes. Okay, cool. Okay, so now it's up and running. So thank you for your patience. Yeah. So why do we study nematodes, right? So this is a picture of a northern root knot nematode that my postdoc took. It's a great image. So why do we study them? Well, they're the #1 cause of crop damage by pests globally. Once you have them, it's a problem. I think of them as like little dogs, right? If you have an adult in a room with a lot of little dogs, it's okay. They may nip at your heels a little bit. It's fine. maybe, you might trip and fall, so it can cause vineyard decline. But when you replant into one of these, it's like putting a baby in a room with a whole bunch of little dogs, right? That could be a problem. And so that's what we try to avoid. And Inga has done a lot of amazing work looking at the effects of nematodes in Washington vineyards and looking at kind of, the influence of rootstock and how that can help young vines grow. So we didn't dig into that, but that was kind of a source of inspiration for these projects. There it goes. So I'm a soil scientist, right? So that was my first joke. And so I'm really interested in how soil properties relate to nematode parasitism. And so part of this is looking at soil health, right? This is a big concept. It's on our list of research topics. And so soil health is defined as the continued capacity of a soil to function as a vital living ecosystem that sustains plants, animals, and humans. And so in a soil that's infested with nematodes that cannot sustain plants that are used to sustain humans, that's an unhealthy soil by definition, right? And soils are incredibly diverse, but again, this concept of soil health, it has to be able to sustain plants, animals, and humans, right? And so this is why we dig into nematodes, is because we want to keep our production up. And so to do this, we took this concept of soil health and kind of decided to probe this like middle column here, right? So if we look at soil health, we can see there's 3 columns to it. We have a physical property or physical component, a biological component. Sorry, I'm trying to get the laser. Oh, there it goes. And the chemical component. So these are kind of like our 3 pillars behind soil health. We have our vineyard in the background, right? This sustains them. So what we did in our experiments is we took the physical pillar. There it goes. So we took that physical pillar and kind of tried to manipulate the physical pillar a little bit to look at the effect on nematode growth or nematode parasitism. And we also kind of played with the chemical pillar. Sorry, to look at this question. Forward. Okay, so here's our overarching question. How do soil health properties influence nematode parasitism, right? So we collected soils from all over Washington and 1 soil from Oregon, and we pasteurized them, and we grew tomatoes in them that we treated with Melodogyne hapla, which is the northern root knot nematode. And prior to growing the plants, we tested the soils for a suite of soil health indicators or properties. Next slide, please. And so what we found from this is that biological indicators of soil health, like permanganate oxidizable carbon, seem to be associated with nematode suppression in the Pacific Northwest. So when we got done with all these experiments and we did a lot of really rigorous data analysis, we were able to start pulling out kind of what was influencing our results, right? And we were able to see that like total nitrogen, total carbon, epoxy, all these things were influential in the response that we were seeing. Next slide. And we took that a little bit further. We used something called structural equation modeling to try to pull out some specific like soil properties that could explain what we're seeing, right? So we might lump like total carbon, epoxy, mineralizable carbon into this like organic matter indicator. And we see that what we find, next slide, is that we're able to explain, next slide. We're able to explain, that went away, some of our results using structural equation modeling. And we were really surprised to find that these like permanganate, oxidizable carbon, total carbon, organic matter cycling type indicators were associated with a decrease in nematode parasitism in our experiment. It was really surprising. And the only explanation that we can give for it is actually from Marcus Kleber here at Oregon State University, who's found that polyphenols, well, and this isn't the only person who's found it. So people have found that polyphenols are, how do I frame this? People have found that nematodes don't like polyphenols. Marcus Kleber has found out that POC is basically a measure of polyphenols in soil. So Ray Weil, who developed this indicator, was like, oh, this is a labile carbon indicator. Marcus Kleber, a few other people were like, no, it's not. It's actually a measure of polyphenols in soil. And so what we're seeing is that with more polyphenols in the soil, the nematode parasitism goes down, which is, there's some mechanistic explanation for that, but it's still pretty surprising. Do you mind going to the next slide? So I made some maps of this, and this is what a map of our nematode, this is the distribution of our soil samples, and each dot represents a vineyard. If the nematode parasitism for our tomatoes was not influenced at all by soil properties, that's what this map would look like. Do you mind going to the next slide? So this is actually the map, right? So as the circles get bigger, we have more nematodes per root. As the circles are smaller, we have fewer nematodes per root. So we see that there's many orders of magnitude difference between the smallest circles and the biggest circles. So that means that there are soil properties that are having a direct influence on nematode parasitism in this system. Next slide. And so this would be close to where I live, around Prosser, Washington. And so we see a lot of these soils are very similar. They're coarse textured, high pH, but we still see major differences in northern root knot parasitism, northern root knot node parasitism in these soils. And so we tried to figure out, we decided to map them against other soil properties. Go ahead, next slide. So it seems like in eastern Washington, pH may play some type of role in northern root knot nematode parasitism. And Inga, Paul, Jungmin, Catherine, and I just published a paper that suggests that low pH soils may have more nematodes in them than high pH soils. We don't really see that here, but so there's something that we've found results that kind of we found results that are aligned with this, but not all that strongly. What we see is that there are other soil properties that just have a much bigger influence on what we're seeing, and that's permanganate oxidizable carbon. Next slide, please. And then next one, please. So here's our POC, right? Sometimes you can't get a lot from these maps, but one thing you'll see is you can't see any yellow. There's no yellow circles, right? And that's because they're all tiny. They're really small. And so this was our nice indicator beyond just like the structural equation modeling that we did, but actually when we map it out, we get similar results to our structural equation modeling. Next slide, please. And we also see it locally in eastern Washington. Soils that have higher permanganate oxidizable carbon typically have fewer, or the nematodes are less successful parasitizing plants in those soils. Next slide. So we did all this work. It was a lot of effort. Catherine, Brandon, my team went out and sampled all over Washington and Oregon. We just published a paper looking at the effects of pH and soil texture on nematode parasitism. We're working on getting this paper out right now where we're looking at this kind of broader scale, more, we're looking at a broader scale study. There's still a lot of work that remains to be done. So the funding that we received from Northwest Center was to develop an automated nematode counting platform. We're still working on it. So where we're at right now is our nematode counting platform is about as good as an undergraduate. Right? And it's interesting because we'll like look at the pictures and we'll be like, I made some mistakes, but it's like makes different mistakes than the undergraduates that we employ. So it's like, that's pretty good, right? It's about as good as like an undergraduate human, but we think we can do better. And so we're working on that right now. It would be great to like expand this. Like the POC results are really interesting. They're really weird. And we can't explain it, but it'd be interesting to like take a set of soils, increase permanganate oxidizable carbon in them, and see if that played a role in nematode success, right? That would be really cool. And so we just need to keep evaluating kind of these biotic versus abiotic components to nematode parasitism. And finally, in the future, I would love to do some X-ray CT work looking at the northern root knot nematode parasitism in the roots in real time. It'd be really cool. And so with that, sorry, I was a little scattered, but I'm ready for questions. Yes, sorry.
I was going to say, Terwieser Hanks at Breeding Insight is also working on that nematode counting platform.
Oh, really? Okay.
So you might want to compare notes.
Okay.
Yeah. And actually, it's a three-part question if you got time. The other part was, are there any practical implications of this? Like managing the per megan.
Okay, so these are great questions. I'm just going to speak loudly.
Yeah.
Maybe I should actually take that back so people won't mind. Hear me, and I'll just repeat questions. Okay, so the questions were, did I know that Breeding Insight is building a platform for something similar? I did not. I talked to Ed recently, and anyway, did not hear that. But there's a USDA, another USDA initiative, the Blackbird Imaging Initiative. I have been in contact with Lance about using their platform for imaging. It's a different style platform. We have tried using well plates before, and we do get adhesion of nematode eggs to the edges of the well plates, which can make counting tough, right? The cool part about the Blackbird imaging platform is it has multiple focal planes, right? And it smashes them all together. You could also take that Z-stack and start doing like, oh, volumetric information about nematode eggs, shape, all these other things that are currently, all we do is count, right? But there's so many other things, like things that we can look at. In terms of the POC stuff, you know, it might help people who are making decisions around purchasing land or other things. I mean, I wouldn't like take it quite that far at this point, right? But it's like, hey, like if you have if your soil health indicators are good, like you have maybe more resilient soil. I think that's like a, you know, a common thought. And so this is just like another case of, you know, these soil health indicators may have real meaning, right? Just, you know, there's a lot of indicators that people find out there. There's often no mechanisms for how they help with soil health, you know, and we're just starting to get to that. Any other questions? Okay, I'll hand this off. Thank you so much.
Thank you, Devin. All right, thank you to all our presenters. We're going to have a quick break here. We'll be back at 3:05 in Ballroom C. So that's where we started earlier this morning. So we'll see you all there. Thank you.